Okay, so for today's lesson, for this lesson, we are going to look at this fluid mosaic membrane. So the membranes that we have, the cell membrane is termed, okay, or is described as a fluid mosaic membranes, okay, by a model called fluid mosaic model. Okay, so what we're going to study here is we have to describe the fluid mosaic model of the membrane with reference to the hydrophobic and hydrophilic interaction that account for the formation of the phospholipids bilayer and also the arrangements of proteins on and or at the membranes. Okay, this is the first part. Second part, we're going to look at the arrangement of the cholesterol molecule, glycolipids, glycoprotein, and cell surface membrane. In the cell surface membrane, including the proteins. We're going to look at this. And we need to know the role of each of them: phospholipid, cholesterol, glycolipids, protein, and glycoprotein in the cell surface membrane. Particularly, we refer them to stability, fluidity, permeability, transport, cell signaling, and cell recognitions. So a lot of things we're going to learn under this part. Okay, for today we only enter part 1. We're going to have the part 2, okay, on the next lesson. So membrane structure that we have learned in the organelle is very, very brief. Now we go into the detail. As we know that the plasma membrane, also known as a cell membrane, is a biological membrane, separates the interior of the cells from outside environment. So if you want to view the cell surface membranes, particularly we want to view this phospholipid bilayer, we have to use an electron microscope because they have high magnification, okay, at least up to, okay, 100,000 times, okay, up to 100,000 times, okay, and we do know that electron microscope have higher resolutions. Therefore, we will be able to view the hydrophilic heads and hydrophobic interior of the membrane. And we do need to know that highlights the bilayer membrane is about 7 nanometer. So light microscope impossible to resolve. Okay, yesterday or previous lessons, we have learned about the resolutions of the microscope. The resolution of microscope impossible to resolve, okay, for us to see as 2 lines. Why? Because the distance between these 2 lines is 7 nanometer, which is less than the resolutions value, okay, the half of the radiations, okay, the wavelength of the radiations of the visible light, 200 nanometer. Therefore, we won't be able to see as 2 lines. We only can see as 1 line. If you want to see as 2 lines, then we have to use the electron microscope. So you can see the electron micrograph, the TEM, transmission electron microscope. You can see as a 2 lines. So these 2 lines actually represent the head of the phospholipids molecules, and then the tail will appear as the pale regions, okay, in between these 2 dark lines. So what are the components of the plasma membrane? The component of plasma membrane includes the major one is the phospholipid molecule. It's a major component, followed by the cholesterol, glycolipids, glycoproteins, and also protein. So in this chapter, we are going to look at the functions, okay, and as well as their structure, okay, and how they actually contribute to the functions of the plasma membranes. So the same diagram appear again. So I stop the recording a while so that you. So assume you guys have done the labeling. So we look at the first structure. So you can see that the short fragments of the sugar. Okay, this one on proteins, this one on the phospholipid molecule. So we do know that one is glycoprotein, another one is called glycolipids. Okay, and we do have the proteins, right? You can see the many protein. So some protein actually embed. Okay, can you see that? Some protein embeds at the membrane here. So we do have what we call the, okay, integral membrane protein. And some of them actually, they do not embed themselves, okay, in the phospholipid bilayer. This kind of protein, we call them as a peripheral protein or a peripheral membrane proteins. Okay, peripheral membrane protein. Those that embed themselves, this membrane protein is known as the integral membranes proteins. Okay, and then we do have this structure. pointing to this small molecule with 4 ring structures. Can you see that 4 ring structures? So this one is termed as the cholesterol. And then we do have the transport proteins to increase the permeability, or do have what we call the selective permeability for the membranes. Therefore, we name it as transport protein. It can be a carrier protein. It can be the ion channel or channel protein. Okay, and we do know that you can see that we learned about this in chapter 1. We have the strand of the protein that attach to the membrane to change the shape of the membranes, right? The cells. So this one we call as the cytoskeletons, which is microfilaments. Okay, microfilaments. And as usual, in this case, we need to know these are the phospholipids. Okay, by layer. Okay, formed by the phospholipids molecules. Okay. So we do know that cell membranes, they are selectively permeable. Okay, understand the word selectively permeable, not semi-permeable. From now onwards, okay, for your A level, always remember the permeability that we describe the cell membrane is selectively permeable. Okay, so how to say that selectively permeable? Because of the interior interior core of the phospholipids actually occupied by this hydrophobic fatty acid chain. So because they are hydrophobics, when the word phobic means phobia, so it means that they're actually impermeable to polar water soluble. So impermeable to polar water soluble molecule, and also the charged ions. Okay, ions, those charged ions, they are not able to move across this phospholipid bilayer. So definitely not by not by diffusion methods. And for non-polar molecule, for example, our oxygen, CO2, as well as lipid-soluble molecule, for example, glycerol, for example, vitamins A, D, E, and K, the ADEC, those are lipid-soluble molecules, so they can diffuse across the phospholipids bilayer. And what is the main thing that causes the molecule? Why molecule can pass through, okay, the phospholipid bilayer based on by diffusion is because of the fluid mosaic model. If you look at this fluid mosaic, basically means that these membranes, they are not rigid. Okay, even though we tightly pack our phospholipid molecule, this phospholipid molecule, they are not stagnant there or non-motiles or non-movements. They still move. Okay, so how they move? They actually can vibrate. You can see that because of the kinetic energy, they can vibrate and diffuse across their mono layer. So from one region to move to another region. So when they vibrate and move, can you see that they create the pore? So this pore allowed the non-polar hydrophobic particle to pass through. CO2, oh, the pore pass through. Can you see that? So this features is known as fluidity. So membrane is very fluid. It's very elastic, very fluid, okay? Because of the movements of these phospholipid molecule create the pore or the gap, a small gap, allow the non-polar particle to pass through. So that's why non-polar particle can diffuse across the phospholipid bilayer because of this pore. But why polar and charged particle cannot? Because of this region. This region is the hydrophobic regions, right? Hydrophobic core. So this hydrophobic core, we're going to repel. Are you clear? You try to imagine that this part is your enemy. Right? They don't like you. Will you be able to pass through this region? No, you won't be able to pass through this region because your enemy is here. They are hydrophobics, means that they don't like water. So any water-soluble ion, they cannot pass through. Polar molecule, they cannot pass through. Why? Because this region is the hydrophobic core. Okay, hydrophobic core act as the barrier. And occasionally you can have the flip-flop. Okay, the flip-flop here is very, very rare. Okay, why very, very rare? Because the head of the phospholipids, they are hydrophilic. Always remember the head is hydrophilic, the tail are hydrophobic. So for the hydrophilic head to move from one regions to another monolayer, they have to pass through the hydrophobic region. So in this case, we do know that unless kinetic energy is very, very high, if no flip-flop is very, very rare. Are you clear? So the movements of the phospholipid molecule contributes to the fluidity of the membrane. And how about mosaic? Mosaic is something like pattern, right? So the arrangements of the proteins give us the mosaic pattern. And this fluid mosaic model is proposed or was proposed by the Singer and Nicholsons in 1972 when they tried to explain the plasma membrane. And the fluidity of the membrane is very, very important. If let's say no fluidity at all, very rigid, you try to imagine we pack our phospholipids so nice, can the CO2 diffuse across? No. O2 diffuse across? No, cannot, impossible for the particle to diffuse across. Are you clear? But our membrane is not true, it's not rigid, they are fluids. Okay, they have a certain degree of fluidity. So this fluidity allows the non-polar particle to pass through. Okay, so this highlight this part. Question always asks about the explanation of the fluid mosaic model. So how we explain. Okay, so the fluid mosaic model describes the structure of the plasma membrane as a mosaic of the component. So what are the components contribute to the pattern? What are the components that contribute to the pattern? So including the phospholipid, cholesterol, proteins, and carbohydrates that give the membrane a fluid character. And the lipid bilayer represent the fluid parts of the fluid mosaic model. Okay, the fluid parts. So lipids bilayer, or what we call the phospholipid bilayer. So the individual phospholipid molecule can move about within their monolayer laterally. So left, right. Okay, vibrate. Okay, by diffusion. And we do have a spontaneous transmembrane lipid motion, or we call it as a fluid flow, extremely slow and rare because we need to have high kinetic energy so that they will be able to move from one monolayer to another monolayer. Okay. So the various protein found embedded in the membranes will give us or account for the mosaic part of the model. Okay? Get the idea what's mean fluid mosaic model. Okay. So with this, I've done for the first part of the lesson.